What I will be telling you today is a lot of what we do in the lab, actually. There is a bit of unpublished data in there as well. And although we work on human embryonic stem cells, I think most of what we do is quite extendable to human-induced pluripotent stem cells, or iPS. So I have no conflicts of interest. I have no shares on the VOB or anything like that. So human embryonic stem cells or human pluripotent stem cells, they hold this fantastic promise that they will be able to do a lot of good things for us. We think about using stem cells, like the first thing in our heads is like the obvious thing, using this in regenerative medicine. So differentiate your cells into some cell type that you need and transplant this to the patients. But there's another more, like, it's actually a booming topic at the moment, is using the stem cells as research models, is using them, for instance, for drug screening or early development or development going wrong, things like that. What you can do is actually making the cell type that you want to drug test and just put your drugs on top of it. It's, for instance, very handy if you're working on monogenic disease where you need a cell line with a genetic abnormality and then you want to drug test for the specific genetic abnormality. You can differentiate the target to the target cell type and do your modeling on it. So, regenerative medicine, it's really immediately going into a fantasy. What can you do with stem cells? You can repair, you can replace organs, so you could, with scaffolding and stuff, keep making kidneys and things like that. You can do a restore of function, something that got lost, like walking. You could restore it again, and maybe even regenerate certain organs again. In fact, since we have stem cells in the world of science, up to now, we haven't been able to fulfill much of these promises yet. But they are now we're slowly getting there. They are the first attempts to move to the clinic. And the first clinical trials are now ongoing, using embryonic stem cells to make retinal sheets that are implanted in the eye to replace damaged retina and to restore, with the idea of restoring sight in very specific forms of macular degeneration. So it's not like for everybody, but it's the first steps. We're bit by bit getting there, but it's been very slow. And one of the reasons is that there's still a lot of practical problems around human embryonic stem cells and human pluripotent stem cells. We have practical problems, really like lab-related problems, and we have safety issues. Still culturing, like bulk culture of stem cells is still pretty hard. Now, if you're going to make something small, like a retina sheet is relatively small, it's okay, you don't need so many cells. But if you're going to make a kidney, it's like a pretty big thing, or you want to make a liver, it's even more. So you need a lot of stem cells to actually start making these big organs, for instance. So, and it is still quite difficult to bulk culture human embryonic stem cells or human pluripotent stem cells. The differentiation protocols are another problem. So there are a lot of protocols published out there. Most of them actually yield mixed cell populations. So it's not that when you differentiate to have insulin-producing cells, you only have insulin-producing cells in your preparation there. You often have a lot of other stuff in there, which you have to start getting out because certain things you don't want to have in there. And they're also very difficult to reproduce. So you download very happily a Nature protocols on differentiating to something, and you try that in the lab, and 2 years later you're still not there, although the people that describe the protocol say that it's got a 90% efficiency in getting, beating cardiomyocytes or whatever, things that seem to be very simple actually now are not that simple. So these protocols are still, far from perfect. And the third problem is that we have not a very good idea about how dangerous it is to transplant stem cells or stem cell derivatives. They have an unknown potential to generate tumors. And it would be a rather undesirable side effect to stem cell therapy to develop a tumor. So we know that stem cell transplantation can lead to tumor formation. It's not very broadly publicized or reported. Actually, I know of one paper on this. This is a case in which a child got 3 transplants of neurostem cells, so fetal neurostem cells, in some clinic somewhere, it's not necessary to mention where, but this was a bit of a despair act for the parents. They went with their child to try to fix the problem. It got the transplants, went back home, developed headaches, and ended up at the hospital. And they found that the kid had developed different tumors in the central nervous system and when they did the surgery, they could see that these tumors actually originated from the donor. So they were female tumors when actually the child was a boy. So, and then they did fingerprinting, and you could see that actually were the stem cells that were transplanted to the child that generated the tumor. So we know that stem cells can form tumors. We just don't know about human embryonic stem cells and how big this risk is. I think I hear air conditioning, right? Because I'm like, it's getting pretty warm here. So from the last 10 years, I know that the change sounds very quick off topic, but I will get back to this. We know that human embryonic stem cells, and now more recently human pluripotent stem cells, they become genetically abnormal in culture. We know that they have a tendency of accumulating aneuploidy, so either whole chromosome or part of chromosomes. Generally it's gains, and there are a couple of them like trisomies 12 and 17 that are very recurrent. And one would ask himself, well, why would this bother us? Why is this a problem? You know, if you pick them out, maybe you just toss them and go on. But actually, one of the hallmarks of cancer is genome instability. You have a lot of traits in cancer that are actually already quite similar to what stem cells can, like for instance, it limits replicative potential, they're immortal cells, and on top they appear to be quite unstable in their genomes. So my work revolves a bit around answering a number of questions that raise on the genome instability. One of them is, to which extent are human pluripotent stem cells actually genetically abnormal, just not only a trisomic or a monosomic or whatever, but how far does this go? How do these abnormalities appear? Where are they coming from? How does it happen that they take over the culture? Because it's one cell that becomes mutant, but in a very short time, you have a whole cell culture that is abnormal. And maybe not always, but it happens pretty fast. And more importantly, when you're going to the clinic, how does this affect the behavior of the cells, especially when you're going to differentiate them and transplant them into a patient? So on the question, to which extent are they abnormal? So there has been already there's a lot of reports on trisomies and, well, mostly trisomies in human embryonic stem cells. But the later studies, so at the beginning everybody was doing G-banding. It's still very common to do a G-banding on embryonic stem cells. But we're moving more and more to higher resolution methods. We're using array CGH basically, which allows you to detect much smaller aberrations than you would if you do a G-banding. If you do a karyotype, you see relatively big chunks of DNA missing or duplicating. From these studies, it's become obvious that about a quarter of the stem cell lines worldwide, they carry a gain of 20q11.21. This is a highly recurrent abnormality, and we know it's the last study, the AMPS paper is a consortium paper of a lot of stem cell labs all over the world who send in samples, and like a quarter of the lines, whether they were iPS or human embryonic stem cells, they carry this abnormality. Most of the labs were not even aware that their lines were carrying this abnormality. We send in some of our lines, but we knew, actually, we already had spotted that they carried the abnormality. Then there's another form of genetic abnormality, and is low-grade mosaicism. It's not that your cell population is always homogeneous. Also, even when you have these chromosomal abnormalities, it's not always all the cells that carry the abnormality. It's sometimes a subpopulation of your culture. And if the cells that are having this selective advantage because of the mutation, they are really very growing faster, for instance, they will take over. But you have a variability in your culture. And this low-grade mosaicism at the beginning has been studied by, there's two papers, the Peterson and the Naftali. They did fish, mostly they did fish, so interface fish on the stem cells. And what they report is that if you compare, you have your lymphocytes and then 2 of the NIH stem cell lines, if you look, the human embryonic stem cells would have about 2% of the cells carrying a monosomy. So it's something that if you do, for instance, a G-banding, you're likely to discard because you say, well, this is, probably an artifact, or it's a too low frequency. On top, these monosomies don't necessarily have to be always the same chromosome. It's just, happening randomly. And they report actually not a very high level of trisomy. This is from the Peterson paper that Naftali has quite similar results, and this is done by fish. Now, in our lab, Kurt has been doing similar type of studies, but we went a bit further. We used array CGH. So single cell array CGH, we look at the full genome of one single isolated stem cell. We did stem cells, human embryonic stem cells, at 2 very different passages, so 300 passages of difference in between of them. We did human fibroblasts, amniocytes, and we looked at, how the type of abnormalities we would find around. In fact, this mouse is really annoying. This is the somatic cells. This is the human embryonic stem cells. And what you do have mostly is not these monosomies that the group of Peterson or the Naftali were reporting, but what you have is segmental gains or losses. So you have parts of chromosomes that are gained or lost, and they are very unique events. It's not like they're not really clonal. So it's a really low-grade mosaicism that is going on there. You have, in fact, in the early passage of our stem cells, we had about 70% of the cells being abnormal. The late passage only had 3% of abnormal cells, fibroblasts again 17%, and amniocytes 8%. In fact, it's a very common thing happening in cell cultures, and human embryonic stem cells are no different than other cells in that sense. There is a significant population in there that is genetically abnormal. But we were quite intrigued on the fact of why we didn't see all these monosomies that had been reported by 2 other groups, by fish. So we started doing fish as well. And we did fish using 3 probes, so 2 telomeric and a centromeric we took randomly 2 chromosomes, so we took 18 and chromosome 1, just because we liked 18 and 1. There's really not a specific reason. And the advantage is that actually you can see, for instance, like here you have the 2 centromeric signals, 2 telomeres P and 3 Qs, and then you see that you have a gain of Q. But what you also see, and you see this relatively often, is actually that your centromeric probe is overlapping. So you have 2 spots for one, 2 spots for the other, so 2 Ps, 2 Qs, and the centromeric probe overlapping. And these cells, they are not monosomic. It's just you have an overlap of the centromere. It actually only happens in embryonic stem cells. For some reason or another, in interphase, those chromosomes, they seem to be very close to each other, or the centromeric regions seem to be close to each other. And when you do a fish, you miscall, and you think this is a monosomy. So, sorry to say, those other guys were wrong. They could not have known, because they did just 1 probe fish. They used the centromeres, and they called a lot of monosomy where there was no monosomy. So, from the data from the fish, we again see we saw actually similarly that we had a lot of loss of telomeric regions, very few monosomy trisomy, but what we did see was actually that the human embryonic stem cells, they have a much higher level of subtelomeric region gain or loss than the somatic cells. So there, when you looked at the array CGH data, they looked pretty similar. Actually, when you look at the fish data, you see that they're different. And what they have actually is a form of subtelomeric instability. The subtelomeres are not mapped on the array, but you do find it back. You do detect this with subtelomeric fish probes. So this has been reported already in mouse embryonic stem cells. So again, human embryonic stem cells in that sense, and they show a quite high level of subtelomeric instability. So next, we are working as well in the lab on epigenetic fluidity. We're working on the X chromosome inactivation patterns as the work of my colleague Mieke Geens. But in the general, I have to say, there's been a lot of studying of epigenetic changes, epigenetic patterns in human embryonic stem cells. And although there is a lot of fluidity and there is a lot of things going on, they don't follow a pattern. It's very difficult to pinpoint, You do see that in timing culture will change. For instance, here you have promoter methylation changes. Early passage is in white, black, late passage is in black. Some lines will gain methylation, other lines will be just the same before and after. And this is actually in the AMPS paper, the consortium, they did a lot of effort in trying to find patterns in these methylation changes. The moment we don't manage to find any. They also show imprinting, but it's variable. Some lines will have loss of imprinting, gain of imprinting. There's all kinds of things going on there, but it is not really following a pattern like in the chromosomal gains that we see taking over the culture. So on this part, we can conclude that human embryonic stem cells and pluripotent stem cells in general, they very frequently become chromosomally abnormal. The whole culture becomes abnormal. They are very mosaic. They have predominantly small genetic changes, like deletions or gains, especially in the septonumeric regions, and that the epigenetic changes are in fact random, that we are not able to find a pattern in them. On the work on the new forms of genetic instability, we've been working together with Lindsay van Houten on the mitochondrial genome. And as you know, each cell contains many mitochondria, and each mitochondria contains many copies of the a little genome, a little circular genome. It's only 16 kb, but it exists in multi-copy inside of each mitochondria, and each mitochondria is in many present in one cell. And they're responsible for the oxidative phosphorylation, but they also have roles in apoptosis, for instance. Now, human embryonic stem cells, they contain very few of these mitochondria, and they are immature because basically they live in conditions of anaerobic metabolism. They don't do oxidative phosphorylation. They only do the first steps, and they don't use the mitochondria for energy. So what we did was actually studying half of the mitochondrial genome with a long-range PCR. It's generating a 8.7 kb fragment. It's a PCR. This is spanning the common deletion region, so it's a hotspot for mutation, in fact, in the mitochondrial genome. And when you do the PCR and you run this on an agarose gel, you do get these bands here. These are just ordinary blood samples of volunteers. I'm also in one of these lanes. It's an 8.7 kb fragment on the top of your gel. When you do stem cells, you do get this. So you have your wild type molecule as it should look, and then you have a lot of little bands appearing all over your gel. So when we first saw this, we thought, okay, we're having artifacts here. This is not normal. But the other possibility would be that these are shorter PCR fragments that are actually originating from the fact that there is a deletion in the region that you are amplifying. So we did excise bands out of the agarose gel. Here you have 6 of these deletions. So this is the same DNA sample. It's repeated several times. You see that there is quite some variation from PCR to PCR. So this is each one reaction. And we band excised 6 of them. We carry out Sanger sequencing, and we do find the breakpoints. So these bands that are here, they really are representing deletions in the mitochondrial genome of the embryonic stem cells. So we screened quite a bunch of cell lines in our lab, and we found deletions in all of them. We found one of the lines that carried at least 7 different deletions. I said at least 7 because we were able to sequence 7 breakpoints. There were many more bands, but you can only say for certain, okay, the ones I sequenced I can say for sure. So at least 7 different deletions in one stem cell line, and at least as soon as passage 3. So it's very early after derivation that those deletions are already present. We did as well as trying to quantify this. As I said, mitochondria are multi-copy, so you have many mitochondrial genomes in each mitochondria, many mitochondria per cell. It means that it's not like the nuclear genome where you have 1 copy per cell, you have a lot of copies, and that's why you have something that is called the mutation load. So you need to go above a certain threshold of load of a certain mutation to get a disease in an individual, so in a patient. So the amount of mutation matters to see whether it will have an impact on the cells or not. What we did for this is single molecule PCR. This basically is taking your DNA sample, diluting it massively until in your PCR tube you only have 1 DNA molecule. And what happens is you have a lot of failure. You basically have all these empty lanes of your PCR, and once in a while you have one that is giving you a signal. And this is what you actually want. You want to be completely at the limit of the technique and just picking up one molecule. And when you do this, you get, this is the wild type and these are deletions. And then you can really count. It is like you're testing molecule by molecule like this. So you count here, you have 4 deletions, you have 15 wild type fragments, so you count a mutation load of about 20%. We did this several times over different stem cells at different passages, and it is more or less about this, what we came out, between 10 and 25% of mutation load as early as passage 3, up to passage 350 that we tested. It doesn't seem to change much. So they're there, and they're in random drift in these stem cells. Next question we ask ourselves is, well, what happens when you differentiate the cells? Because human embryonic stem cells, they are anaerobic. They don't use their mitochondria to make energy. Maybe they don't give, they don't have a problem. I was going to say something ugly now. They don't mind about what's going on with the mitochondrial genome. But maybe when you differentiate them and they need suddenly this mitochondrial genome, it's becoming a problem. So we took the undifferentiated stem cells. Here you have example of 2 of our lines, wild type, deletions. This is the undifferentiated state. You differentiate them and they look exactly the same. So these mitochondria, they are still functional enough to undergo cell differentiation, and your mutations are still present in the differentiated progeny of your cells. So, conclusion is that human embryonic stem cells, they carry a lot of large deletions of their mitochondrial DNA. The load is about 20%, and they're still capable of differentiating, and after the differentiation, they still carry the deletions. We have no idea what impact this has for the clinical use. We don't know if human iPS actually carry the deletions, and we have no clue where they're coming from. We don't know anything about the origin. So this is a lot of work that is still ongoing in the lab. So more work of Kurt. He's been working on how the chromosomal abnormalities appear in the culture. So it did catch our eye that the stem cells carry a lot of DNA like gains and losses that are small regions or half a chromosome. It's not aneuploidy, which you can explain by anaphase lag or non-disjunctions or things like that. It's DNA break that has to happen there. And actually when you start looking at the breakpoints, when you do array CGH, you see that there's a bias towards common fragile sites. These are sites of chromosomes that are easily breaking, more easily breaking depending on the culture conditions, and that immediately makes you ring a bell because, yeah, they're cultured cells. So what did we do? We made up an hypothesis in which we said, okay, there is something going on in the culture, the way we culture the cells, that is inducing DNA breakage, and this is leading to all these chromosomal abnormalities that we see. So medium depletion in high-density cultures leads to either a shortage of nutrients or an excess of waste. Because they have the cell cycle that doesn't really check what's going on before going into S phase, they just go for it when it's actually not going very well in the culture. This leads to replication fork stalling, so your DNA replication fork gets stopped. Your DNA ends up breaking, you have double-stranded breaks, they end up being chromosomal abnormalities. So this is what we thought. 4 years of work later, can happily come and explain the results. So what did we do? Kurt amused himself by culturing human embryonic stem cells at different culture densities. We go from a very low density, so this is it's a mouse feeder layer, so this is how we commonly culture them. It's like shifting culture systems, but this used to be 4 years ago how we commonly cultured them. Very few colonies. So when we are looking at a dish like this under the microscope, you think, oh my God, the cell line is dying. It's going really bad with the culture. We're never going to be able to do an experiment because this is very low density. This is a density you're thinking, okay, it's going well. I'm expanding the cell line. Soon I'm going to have enough cells to do an experiment. This is a normal dish. This is like, oh yeah, okay, it's going well. And you keep the cells culturing like this. And this is a very high density. So when you're culturing like this, is actually, you're overdoing it a little bit. There is too many cells in one dish. We measured DNA damage using 2 assays. One is counting foci of the phosphorylation of the histone 2AX. And that's a typical marker used for double-stranded breaks in DNA. You can do this, it's an immunostaining. And we did the common assay, which is very nice. It's actually you embed the cells in agarose and you pull the DNA out with an electric field, just like you would do an agarose gel. And the longer the tail of your comet, the more DNA damage there's been, because it means that the DNA is chopped in little pieces and you can actually pull it easier out of the nucleus than if it's, yeah, I'm sorry. But you guys also took longer. Huh? Okay, so I'll tell you about this and I'll skip the other one, okay? I'll finish this one and... Anyway, results. So we had the low density, and this is the high density, and this is the normal density. So what you have here is the comet assay, you have an increase of DNA damage, the higher density, but at normal culture densities, actually you already have a significant levels of DNA damage. The counts of a double-strand break foci, they actually give the same. So the higher the density, the more the DNA damage, but also again, at normal culture density, quite a lot of DNA damage going on already. And if you look at the groups, you actually, what you're seeing is that there are more cells with a lot of damage actually in your culture. If you do array CGH, again, single cell array CGH, what you see is in low density cultures, and this is actually only one passage at low density compared to one passage at high density, you go from 3% of normal cells to 36% of normal cells, so it's an enormous impact on chromosomal abnormalities. And when we started looking at the culprit of this is by culture media analysis, you have a decrease in glucose, but this is not a very significant one. You have no depletion. You have a decrease of aspartate and glutamine, which are amino acids that are involved in the nucleotide synthesis. But also, it's significant, but it's not very biologically meaningful. But the lactate is going up, and the lactate is going up massively, and your pH is starting to going down. So what the cells are having is damage because of medium acidification. You can mimic this. We did this by adding active lactic acid and by adding acid. So taking cells that were grown on very low density, adding the lactic acid as if they would be in high density, you basically have, we basically prove that it's the lactic acid in the medium that is causing the DNA damage. And if you change the medium twice a day instead of once a day, you have a rescue. So you basically, if you want to culture your cells at high density, you have to change the medium much more often so that it doesn't get that acid. And if you look at the DNA damage, it's exactly the same. So you get every time that you change the medium twice a day instead of once a day, you get a decrease of chromosomal abnormalities in the culture. So conclusion on this is that high-density cell culture is leading to accumulation of lactate. It's acidifying the medium massively, and it's leading to DNA damage. And this is going to the chromosomal abnormalities. Well, you guys have all the slides of the selective advantage, so I'm sorry, but I will have to go over them. I'll just go to the end. We have not done this work yet, but this will be something we would like to do, is what happens actually with these abnormalities on the behavior of the cells. There are already a bunch of papers telling you that aneuploid embryonic stem cells differentiate worse than euploid embryonic stem cells, although there has been no systematic study on what is happening for each type of abnormality that you have, how this is affecting differentiation, and how many abnormal cells you need to have a problem. When you look at tumorigenicity, as I said, there is a lot of similarities between human embryonic stem cells and cancer cells. So maybe just a few changes are enough to trigger quite a bad phenotype in these embryonic stem cells. And if you look at this here, you have the gene expression of euploid embryonic stem cells, aneuploid embryonic stem cell derivatives, and astrocytomas. I'm done. It's the last one. So you can see that the heat map for the aneuploid embryonic stem cell derivatives is much closer to the astrocytoma than it actually is of the derivatives of the euploid line. So I will let you to read the take-home message yourself. You have it printed, I think, on your papers. Just that if we think, in the context of the last one, I really want to say is in the context of this making gametes out of human embryonic stem cells. I just told you the cultures are mosaic, the cultures are genetically abnormal, they carry mitochondrial deletions. One human pluripotent stem cell would equal one gamete, would equal one child. So please, take this home and think about it if you're thinking about making gametes out of human embryonic stem cells. Thank you, and sorry that I took long.